Endospore staining technique. So this is an example of a differential stain in that we're going to be using two stains to differentiate between groups of bacteria. So we want to start off with that thick smear that you're becoming an expert at conducting. Remember, 1 drop of water, 1 colony, swirl around and allow to evaporate. For this technique, we want to determine whether we can visualize endospores. So we're going to make use of this boiling water or close to boiling water, as well as this heat station. And our first step is going to add malachite green. So malachite green is this really robust green stain. It's a direct stain, so remember it's basic, positively charged. But the important part of this step is that we want to apply high heat, steam heat. We do not want this, we do not want the thick smear to dry out. We want the dye to be there for the duration of the time that we have the slide in the steam heat, which is 5 minutes. So you have the option of sort of sitting here and watching it and making sure that it doesn't evaporate. If it needs another drop of dye, you can add another drop of dye. Other techniques make use of a little bit of paper towel on top, which sort of holds the moisture in. So you can use either of those options to keep the thick smear wet for the duration of the 5 minutes. So once we're done with that step, and we'll want to be careful because this might be awfully hot. It's not quite boiling for my example, so we probably want to put this on using our clothes hanger. We're going to get rid of the dye. Walk over here. And then we have our second and final step, which is known as the counter stain. Saffron in. Apply the dye on here for 30 seconds. Now, the steam heat allowed the endospores that may or may not be present in your sample, it allowed them to essentially open up and accept the dye. And this counter stain is going to stain everything, whether you're an endospore, actually the endospores will resist the stain, but whether you're a vegetative cell, any other cell will pick up this red dye. So when you visualize, all the cells should appear red, and if you have any endospores, you might see green dots inside of some of the cells, or alternatively, you might see some tennis racket shapes or safety pin-like shapes. If there's green inside of them, that is a strong suggestion that your microorganism has the ability to form endospores.